A very good morning to all of you. So today discuss an important topic, that is ubiquitination and proteasomal degradation. One of the important topic for the short note for your many of university exam. Okay. So first understand what is the process of ubiquitination. First we will understand what is ubiquitination process. After that, how these ubiquitinated protein will undergo the proteasomal degradation. Okay. So what happens first? Suppose if inside the body there is any protein. Suppose this is the protein, for example. Okay. Suppose this is a protein, and this protein, due to any reason, it is inside the body, it's a toxic in nature, or it is non-functional, or it is misfolded. Means this protein is ultimately not utilized by the body. Then what will happen? This protein will not remain as such inside the body, because if it will not undergo any degradation, then what will happen? If it is not degraded and it will remain as such inside the body, then it will cause a number of disorders. So the mechanism is inside the body, the protein which is non-functional inside the body is undergo degradation. In our body, a number of proteins are there. Now the question arises, how this proteasome will detect that which protein should undergo degradation. There is some kind of tagging will be there. Suppose there is a lot of, suppose you are going in a crowd, okay, and someone is want to recognize you in that crowd, then how that person will recognize? By some kind of tagging, or you should have some wearing some different type of color, and you will easily recognize on that crowd. Similarly, what would happen inside the body? Suppose this is the target protein. This is a protein which has to be undergo degradation, because it is ultimately non-functional inside the body. Then what body will do? First, body will tag it. Okay, this is a protein, and first body will tag it with the help of another protein, and that protein is known as ubiquitin. Okay, so remember, ubiquitin is nothing. Ubiquitin itself, it is also itself a protein, but it is a tagging protein. Okay, what is the name of this ubiquitin? This is a tagged protein, or you can say this is a tagging protein. The main function of ubiquitin, it will tag that protein which has to be ultimately undergo degradation by the proteasome. Clear? Because a number of thousands of proteins inside the body, then how body will recognize that which protein undergo degradation? So by seeing this tagged protein, proteasome can easily recognize, okay, this is the protein which has to undergo the degradation. Okay, so this is the basic. Now let's understand how this process of ubiquitination, that is how this target protein is coated or tagged with ubiquitin. Before that, one important thing is that always remember the attachment of the target protein with ubiquitin occur only at the lysine position of target protein. None elsewhere. Wherever in target protein there is a lysine amino acid, from that lysine amino acid that ubiquitin will attach, and it will tag that target protein. Clear? This is the basic thing. Okay. Now let's understand how this process of ubiquitination will take place inside the body. So as I told you, ubiquitin is what? Ubiquitin is nothing, but it is also a kind of protein. Okay. Then this ubiquitination process consists of three enzymes, E1, E2 and E3. Three enzymes in this ubiquitination process. And this E1 enzyme is what? This E1 enzyme is ubiquitin activating enzyme. Okay. What is the name of this E1? The name of this E1 is, this is a ubiquitin activating enzyme. And this E2 is ubiquitin conjugating enzyme. Conjugating enzyme. And this E3 is ubiquitin ligase enzyme. Clear? So with the help of these three enzymes, the process of ubiquitination will occur inside the body. What is the name? There are three enzymes, E1, E2 and E3. What is the name of E1 enzyme? E1 is ubiquitin activating enzyme. E2 is ubiquitin conjugating enzyme. And E3 is ubiquitin ligase enzyme. Clear? As to how this step will occur, let's understand that. So E1 is what? E1 is ubiquitin activating enzymes. As the name suggests, ubiquitin activating enzyme. That means the function of this E1 enzyme is to activate this ubiquitin. Okay, that's why the name ubiquitin activating enzyme is there. Then what happens? Suppose this is the structure of ubiquitin and this is the carboxylic group. This is the carboxylic end of your this ubiquitin. Then first what will happen? This E1 enzyme, that is ubiquitin activating enzyme will come, and this is an energy dependent process. In this process ATP is required. Okay, energy dependent. That's why ATP will provide the energy for this process of ubiquitin activation. Then this E1 enzyme, that is ubiquitin activating enzyme, will come and it will attach with this ubiquitin through this thioester linkage. This is what kind of linkage? This is your thioester linkage. Okay, thioester linkage. So now at this point what will happen? At this point this ubiquitin is activated inside them. Yeah. The first thing what will happen? This is the ubiquitin. This is the carboxylic group of ubiquitin. As I told you before, ubiquitin is a protein, so it having a one carboxylic and one amino terminal also there. Okay. So at this carboxylic end of this ubiquitin, this enzyme E1, that is ubiquitin activating enzyme, will come and ATP will provide energy for this process. Then through the thioester linkage, this is what? SE1. Through this thioester linkage, the ubiquitin is ultimately activated. First step. Second step. In second step, this ubiquitin is what? This is activated ubiquitin. Then the another enzyme E2. And that E2 is ubiquitin conjugating enzyme. That enzyme will come. And it will conjugate with the this E2 enzyme will conjugate with the cysteine end of the ubiquitin. Clear? This is what? This one? This is the activating ubiquitin. On this activating ubiquitin, this E2 enzyme will come, and E2 is what? Ubiquitin conjugating enzyme. So then what is the function? This E2 enzyme will replace this E1 enzyme and get attached with the cysteine portion of the ubiquitin, and now it is conjugated with this ubiquitin. Clear? Now comes on the third step. This is what? This is E2 attached, that is conjugated ubiquitin. Conjugated ubiquitin. In third step, your third enzyme, that is E3 will come, and E3 enzyme is ubiquitin ligase enzyme. Okay. And what is the function of this ubiquitin ligase enzyme? It will form with peptide bond. This is what? CONH. This is what? CONH is your peptide bond. Na? CONH is a peptide bond. Na? So what will happen? This ubiquitin, first this ubiquitin, then attach with the lysine portion of the target protein. Okay, this is target protein, and in that target protein, the lysine amino acid end will attach through the peptide bond with this ubiquitin. This is what? This is the peptide. Clear? Clear? And when it is attached, this ligase enzyme will form peptide bond, and there is attachment of this ubiquitin through the lysine portion of target protein. Then this E2 enzyme will remove, because the function of this E2 enzyme will over. It is the only function of E2 is conjugated the ubiquitin. Up to this process occurs. Then again same process will repeat. Then another ubiquitin will attach. Then again all these three steps will again will occur, and then again another ubiquitin is attached. So attachment of a number of ubiquitin or tagging of a number of ubiquitin to the target protein is known as polyubiquitination. Yeah. Let's understand it again. What is the process of ubiquitination? The process of ubiquitination is to tag the target protein, which ultimately has to undergo degradation through the proteasome. Okay, and the process of tagging of that target protein with ubiquitin protein is known as ubiquitination. And this ubiquitination process consists of three enzymes: E1, E2 and E3. E1 is ubiquitin activating enzyme, E2 is ubiquitin conjugating enzyme and E3 is ubiquitin ligase enzyme. Then first step is that this ubiquitin, at this ubiquitin, the even enzyme will come and with the help of ATP, that is ATP will provide energy, then at the carboxylic end of this ubiquitin, this even is attached with ubiquitin through thioester linkage. First step. In second step, this ubiquitin is what? This is now activating ubiquitin with the help of this enzyme even. Now this activating ubiquitin enzyme is acted upon by another enzyme E2, and this E2 enzyme is what? Ubiquitin conjugating enzyme. Then this E2 enzyme will replace this even enzyme. Okay, this E2 enzyme will replace this even enzyme and get attached the cysteine end, the amino acid cysteine end of the ubiquitin, and this process is ultimately causes conjugation of ubiquitin. In third step, what will happen? This conjugated ubiquitin, then third enzyme E3, that is ubiquitin ligase will come, and the function of this ligase enzyme is what? It will form peptide bond. CONH is what? Peptide bond. Then ubiquitin will form attached with the targeted protein lysine residue of type. Remember, ubiquitin always remember at the lysine end of the targeted protein with the help of which bond? Peptide bond. This is attachment of first. Then again all three steps will go on attachment of another ubiquitin. Again this step will proceed attachment of another ubiquitin. So this process of attachment of multiple ubiquitin to the target protein is known as polyubiquitination. Ultimately our protein which is targeted is tagged with the ubiquitin. Okay, and this whole process is known as ubiquitination. Now this ubiquitination will undergo proteasomal degradation. How this will degrade into proteasomal? Let's understand that also. How this proteasomal degradation will occur. As you know, there are two kinds of degradation. There is a lysosomal degradation also, proteasomal degradation also. But always remember, the lysosome consists of variety of acid hydrolase enzyme and simply will protein undergo degradation in the lysosome. But for the proteasomal degradation, protein must be tagged with the ubiquitin. Okay. So, in most common proteasomal degradation include 26S proteasome. Okay, most important is that of your 26S proteasome. So, the structure of proteasome is like it is a cylindrical in shape. It's like a cylinder. Okay, and this proteasome consists of two units. This 26S proteasome consists of two units. One is 19 unit, other is your 20S unit. And this 19S unit is your regulatory unit, and this 20S unit is your catalytic unit. Okay. So this is the structure. This blue one is what? This blue one is 19S. This blue one is this one. This is the 19S subunit of the 26S proteasome, and it is what? This 19S is your regulatory unit, and this one in between, this is your 20S subunit. And remember, this 20S subunit consists of two parts. One is two portions is that. One is there is a alpha subunit. This red one is what? This red one is alpha subunit of 20S. Okay. And in between this red one, this is the beta subunit. And this is black one is a beta subunit. Okay. So let's understand again the proteasomal structure. First thing you should know, the proteasomal which mostly undergo degradation, that is of your 26S proteasome. And out of this 26 proteasome, it is a cylindrical shape structure, and it consists of two kind of unit. One is 19S, another is your 20S. And in 19S is your regulatory unit and 20S is your catalytic unit. And this 20S and again consists of alpha and beta subunit. Okay. This outside red one is your alpha subunit and inside this black one is your beta subunit. Clear? Now suppose this is the targeted protein. And this protein is what? This protein is ubiquitin. Suppose this is the ubiquitin. Then what is the function of this 19S subunit? Then function of 19S subunit is to recognize this ubiquitin coated protein. Okay. Suppose this ubiquitin tagged protein is roaming here and there inside the body, then which will recognize? This 19S, that is regulatory unit, will recognize, oh, this protein is ubiquitin coated, that means we have to degrade it. Okay, so this function of 19S to recognition of this ubiquitin coated or ubiquitin tagged target protein. Okay, now once it is recognized by this 19S subunit. As it is recognized, then it will what? It will enter inside this 19S subunit. Okay, and once it is entered, then this ubiquitin will be removed. Okay, as soon as that tagged target protein is recognized by this 19S subunit, and then as recognition, it will enter inside it and ubiquitin will degrade it. And this ubiquitin is again utilized for the tagging of another protein, because it's a protein, so it's not, body will not waste it, na? So it will utilize, but it is recycled. It will utilize tagging of another protein inside the body. Okay. Now what happened? Now this folded protein comes under this 19S regulatory unit. Now another function of this 19S is that it will cause us this folded protein. This is what? This is the folded protein. Okay. Because protein is in folded form, whether it's a functional or non-functional doesn't matter. It is folded form, then convert this folded form protein into unfold form. This protein is what? Now it is your unfold form. Unfold form. This is the function of the 19S. Okay. So remember, what is the function of this 19S subunit? First, it will recognize the tagged, or you can say ubiquitin tagged protein. First function, first it will recognize. Second function, it will unfold. It will unfold the protein. The protein which comes in the folded form, it will unfold that protein. Now the protein will open. And this unfolded protein enters inside the catalytic unit. That is your 20S unit. First it will enter into the alpha subunit, and after that it will enter into the beta subunit. And ultimately, what is function of this catalytic unit? It will degrade the peptide bond. And ultimately the protein is degraded in the form of amino acid. Okay, and this ultimately degraded protein gets disposed of through the body. Alright, so this is the whole function, how the ubiquitin will tag the target protein and how the ubiquitin tagged protein undergo proteasomal degradation. So very important topic for the post-graduation student and a short note for university exam. Okay, so this is also one of the must-read topic. I hope you will understand, like, and you will subscribe and share the video. Thank you.